one and all. Myself Dr.J.Hemapriya from Department of Microbiology. Today I am going to discuss about the topic Ti-plasmid. So what is Ti-plasmid? Plasmids are nothing but extra chromosomal self-replicating body which is present in most of the bacteria, and it gives an extraordinary power to the bacteria that we know already. And what are Ti-plasmids? Ti means tumor inducing. Okay, so Ti-plasmids are called tumor inducing plasmids. So Ti-plasmid based vectors are having very high efficiency. That's why we call them as high efficiency vectors. And it was the main aim of developing this Ti-plasmid is to introduce new genes into the plants actually. So Agrobacterium tumefaciens, actually this is a bacteria which is a gram-negative bacteria, and it is a soil bacteria. This Agrobacterium tumefaciens only possesses this Ti-plasmid, and this bacteria actually causes a disease called crown gall disease in plants, in which the cells grows in an undifferentiated manner, because we all know what is a tumor. Tumor means undifferentiated growth of cells in an uncontrollable manner. Okay, right. So which results in the formation of a tumor. So this Agrobacterium tumefaciens, it's a gram-negative soil bacteria, and it possesses the tumor causing ability, and because of the presence of this Ti-plasmid. Alright, so what is the structure of, how do we define the structure of Ti-plasmid? Means it has 196 genes, including one structural RNA which encodes for 195 proteins. And this plasmid actually has a very important region called the tDNA region, which is about 20 kilobase pair in size. And this is the region, actually this is the unique region which is going to get transferred to the plant cell, and it gets integrated into the plant DNA. So this is the most important portion of the Ti-plasmid actually. And even though after the transmission also, the most unique feature of this tDNA is it stably gets transmitted through the divisions, like when the plant cell undergoes mitosis and meiosis. Even after the mitosis and meiosis also, it does not get disturbed, and the tDNA gets successfully transmitted from one generation to the next generation of the cell actually. So that is a unique feature. So this transfer of tDNA from Agrobacterium to plant cell, it depends on the 25 base pair repeated sequence which is present at the both the sites of the tDNA. Actually, when I discuss about the structure of Ti-plasmid, you will come to know what are these repeated sequences on both the sides of the tDNA. Okay, then the tDNA carries an enzyme actually. So it is having enzyme. How it causes tumor? How the Ti-plasmid is having the ability to cause tumor means this tDNA carries enzymes for certain kind of plant hormones which are nothing but the auxins and cytokinins. So we all know that auxins and cytokinins are nothing but they are all phytohormones which can cause the abnormal cell growth actually. So because of this particular reason only, when tDNA enters the plant cell, a tumor is getting formed actually. So it also carries genes for, there are other type of genes called opine genes, because see the bacteria is going to survive in the plant. So how do the bacteria get food? So bacteria needs food also. So what it will do is, it will carry a gene called opine gene which could able to synthesize its own food actually. So this opine serves as a food for the bacterium. So it's carrying its own tiffin carrier, it's like that. So it is taking the opine, there is a opine gene which is going to act as a food for the bacteria, right? So it goes comfortably, cause disease, sit there, eat very nice food, and then it will grow as a tumor. That's what going to happen here. Okay, so and what the researchers have done is they wanted to use this quality of the Ti-plasmid in the genetic engineering purpose. So what they did is they did some change in the Ti-plasmid, and genetically they converted the Ti-plasmid into a transfer for the use of transferring the genes actually, right? So what they did for this particular conversion is they disturbed the tumor causing gene as well as the opine synthesis gene, and they replaced them with the selectable markers which is conferring the resistance to the antibiotics. And Ti-plasmid based vectors were also constructed with the following components. So these are the components actually the recombinant Ti-plasmid has. Okay, origin of replication. So origin of replication is must for any plasmid, that we all know it. So it allows the plasmid to replicate. The second one is, as I said, the tDNA will be accommodated by on both the sides by two borders called right border sequence and left border sequence. Okay. So the right border sequence which is necessary for the transfer of Ti-plasmid. So the right and left border sequence, they are only the most important part of the Ti-plasmid, which is going to help the tDNA to get transferred from the Ti-plasmid, I mean from the bacterium to the plant cell. Okay. And it is also having a multiple cloning site, because we have to insert our own gene of interest, right? So for that purpose, we are going to insert, I mean the researchers have inserted a multiple cloning site also for the ease of insertion of any gene of interest, okay, and between the tDNA border sequences, right? So recombinant genes can very easily get integrated into the tDNA of the Ti-plasmid, and the plasmid can be used to infect the plant cells, okay, right? So the infected cells are placed on a culture medium, because this all of us know how to select the recombinant cells, or how to select the recombinant like culture cells. The infected cells will be placed on a culture medium which contains growth factor as well as the selectable antibiotics, because we have already inserted selectable markers which are nothing but the antibiotics. So we are going to put it in a medium which contains the antibiotic. As a result of that, all the other cells will die, only the Ti-plasmids